like last session. If it is too boring, let me know. We will change a slight track and then come back again. Don't worry. And again, be active like last time. If you have any questions, stop me and ask a question. So that will be more productive. So let me share my screen. Host has to enable to share. Madam, could you please give me
I'm sorry.
permission? No problem.
Please check.
Yes, I can. Thank you. Okay. Now, I want to make sure that two things before we start. Let me check. Still, people are joining. One main thing, I don't want anybody to do or work on the software while I am explaining it to you. At the end of the session, I will share the tutorial PDF file, right? So that it will help you on throughout what I'm doing today. Second, I will also show you how to download. The license files are shared to the organizers. They will share the license file to you. The license file will work for the next 12 days. So it will work for the next 12 days for you. So you can learn, try it out, play it around, no problem. And third, there are some optional downloads, which I'll explain. If you're really keen for that, you download it, otherwise not required. Otherwise, the basic, most of the things can be done with the software itself. The software works only in a 64-bit machine, not in a 32-bit machine. 64-bit is mandatory. And I'm sure most of the machines are now 64-bit. And not requirement of high amount of RAM or GPU cards, nothing much. But one of the very important requirement is your machine should have a graphics driver which supports OpenGL 2.0. So most of the drivers are latest drivers will be fine. Latest drivers should be fine, but otherwise, if it is showing some graphics issues, just try updating your drivers, display drivers. Again, I'm repeating, GPU cards are not required. So then that should work. And otherwise, I'll be explaining different perspectives. So I recommend everybody to be very attentive here. So that will be really helpful. So just a minute. So today we will be going through a software package called as CSAR. The CSAR means C structure activity relationship. So you can visualize and make decisions. Apart from that, there will be supporting factors anyway that will be coming up that we will go through it one by one. The first thing I want to show is that the website biosolveit.de. It's at Germany, and it has came out from Matthias Rere group, where most of the packages have been through an academic collaboration, and also it has been enhanced, and many more developments and feature-rich enrichments have been done from the industry side, and a lot of collaborative work from industry, including pharma industry. Now today we are mainly focusing on the CSAR package. And there is another package called as Infinicy, that is chemical space explorator, where you can design new molecules based upon pharmacophore. CSAR is completely for docking, virtual screening, scaffold fragment-based approaches. Even you can search similar binding pockets with other PDBs. Those all things can be done in CSAR. The components within CSAR is hide, flexex, and record. These are all some other methods not in Caesar, but they are in Infinity. In order to download the package, first you can go to Caesar or you can click on the download. Just click on the download. Then you will get a page like this. As I said, all should be 64-bit. You can download Windows here, Mac or Linux. Here, Linux, no installation, just extract and run it. Windows, there is an installation. Mac also, there's an installation. Once that is done, if you scroll down, there is something called version info recore index. If you click on recore indexes, that's where we need a fragment database. So during the explanation, I was talking about fragment database. So if you scroll down, there are 3 databases available. The first 2 are free. The third one is paid. We are not charging, but Cambridge Structural Database is charging for that. So if you have a subscription for that, you will get access to that database. Otherwise, Zinc and PDB fragment database. It's not the full compound. It is a fragment database. They are non-redundant. Duplicates are being taken care. So they are non-redundant fragment databases. And additionally, what could be downloaded is that here also you have something called FlexX command line on your left hand side. So if you click on that, you have FlexX command line, which will also, I told you, you will be able to do scoring. So FlexX command line will be able to do large virtual screening even for millions of compounds much faster, and also will be able to calculate the docking score for you. So that is how the way you can calculate docking score. Now, being its command line, don't worry. It is just single command, not multiple commands or multiple steps involved. It is just one single line. That's it. And it takes a lot of time if you have a lot of compounds, less time if you have less compounds. Now, the speed of the calculation completely depends upon your number of cores, speed of your processor. Oh, sorry, speed processor, as well as the RAM that you have. So it doesn't depend upon whether the software is fast or bad. It is completely depending upon your efficiency of your machine. Any software, any computational software you take, it all depends upon the efficiency of the software. That doesn't mean that this does not work on a normal laptop. It does work. Of course, it takes more time. The question is how fast you want the things to be done, right? So that is exactly we are looking at. Now, do you have any questions before moving to the software? Oh, let me check the chat. Sorry, I didn't know. I didn't realize that. Okay, no, things are clear. Great. And I hope all of you have the license file and instructions to download. Yeah, by the way, one thing I forgot. So when you click on download, it will ask to register. So let me, yeah, correct. So when I click on this to download, it will also sign in or register. You can click on register. You can give your personal email address or your academic email address, whoever. They don't spam you with a lot of emails, so no problem. And once you register, then you will be able to download the software. So you have to register mandatorily. And I don't recommend you to do it right now. As I told you earlier, you have the license file. For the next 12 days, the software will work for you. Aramse, you can take, after the session, you can start. And I can see that this is already being streamed in YouTube. So you can always go back and look at what are the options that we discussed. That's what I said, Sandesh. Don't open LIC file because that's what the next step I'm going to say. When you open the software without license, the software will work for 1 or 2 days, 1 or 2 days maximum. After the second day, the software will automatically ask you the license. Then you click on license file and then do it. Otherwise, if you think that you want to do it right now, when you open the CSAR software, let me open it again. Okay, I am using Mac. So when you are opening CSAR software, yes, like here. This is how it comes. And then it opens up a front window which talks about like this, a window will come. If you're already working on some certain project, it will show up like this. Otherwise, it will show new project or start CSAR2. Let's go for new project. Click on new project. Then the window opens like this. On your right hand side, top, there is something called system button. Click on that button. It looks like settings button. If you click on Inspirator, that is where we do all the fragment based approaches. In your email, we have mentioned optional fragment databases. If you download those databases, each database is of 1 GB. Okay, keep that in mind. Each database is of 1 GB. You have to click on, after the download, you can have to click on this folder, and then you have to select that zip file, whatever you have downloaded, and then automatically it gets added up here, right? And then you can go back, and you don't have star drop installed, so it will show into mark, that's fine. And also click on RCSB PDB. If you're connected to internet, okay, automatically it will update all the PDB ID codes here. So if you just click here, it will get updated. Mine is till March 12th. So now I gave to update. So it will update all the PDB ID indexing, and automatically software will be able to download based upon the PDB ID that you are able to give it. That is one. These are all small handy tools which is helping you rather than downloading, loading, and all separately going to browser. Just if you know the ID, it will be able to update it automatically. Okay, so this will take some time. Let it do it. Yeah, still going on. Let's see. Depending upon your internet speed, everything, it will take time. So it has to download more than 175 PDBs. I shouldn't have done that because it's taking time. Okay. I just closed it because it is saying that I don't want to waste the time for it and keeping you wait. So I'll open it again. And I'll show you the rest of the data. Okay, so new project and again system. And you, if you have internet proxy for your institution, please give the proxy details here. Now, if you want to install the license right away, click on this license and you can click on select file. Whatever the .lic file you have downloaded, you can click that file and it will open and install. And it will tell you how many days left here automatically. Okay, you need not to click on request license file. Already you have license file, you can just select. Right? And then again on the system, you have, if you want to update, you can update, or if you want to have a readme, some quick readme issues or other things are all discussed here. Now, if you want to go to help menu, you can click on help menu. There you have a beginner's guide, tutorial movies, online help, first start help, and what's new. So this all takes you to your browser. Okay, so ensure that you have internet in your machine. So the whole interface, I'll explain it one by one once we start. But we have different modes in the software. If you go to the middle of the package, the sender region of the package, you have different modes. So let me explain mode by mode. The first one is basically like proteins. If you want to load your protein, if you want to mutate the protein or make modification, flipping the atoms, flipping amino acids, all those things can be done under the protein tab. Then you have binding site tab. Also, binding site tab is where you will be able to define your amino acids, which amino acid to be considered, which amino acid not to be considered. So all those things has to be taken. And also regarding protein editor, As I told you, if you want to modify things, you have to take care of that. Analyzer is something that, let's say you did docking using Autodock or Vina or some other packages, but you wanted to calculate the torsions, you wanted to calculate the binding affinity using CSAR, then Analyzer will help you. Another part with Analyzer is, let's say you are importing 2D structures of your ligand. So automatically Analyzer has a package which takes care of conformation analysis and also ring conformations, and it will take care of 2D to 3D conformation conversion. And Analyzer also has the efficiency to compare between the trend of affinity as well as your experimental data. So it can do a statistical analysis, but for that you require minimum 5 compounds, wet lab experiment data, as well as docked binding affinity data. That can be also put in your publications for that statistical analysis. Then you have molecular editor. If you want to make changes in your structures and then redock and see, that is called molecular editor. Then you have inspirator. Inspirator is where you do structure-based, scaffold hopping and fragment-based drug discovery. And in the docking, you can do a docking generation of poses, virtual screening, binding energy, binding affinity calculations, as well as torsion analysis. You can always do template docking, normal docking, and also you can carry out pharmacophore-based receptor mapping constraints can be also defined. Today, we will more focus into docking and others. Rest of the things, if we have time, we will see. Mostly, we will try to cover it by 4:15, 4:30. I hope all of you will have patience for that. So by default, when you open the software, it is under the proteins mode. Okay. So I'm going to the PDB and I'm giving one STP, which I already know the code. Now, before deciding with the PDB, I already told you in the morning, have a thorough understanding about the mechanism, which PDB you have to choose based upon resolution, lowest value of resolution, also whether the PDB is druggable, whether the PDB is bioavailable, and its quality. So based on, and also if it has a definite functional sites and other things well defined. Then you choose your PDB and just give the PDB ID here, and you can choose one STP by clicking on this. So this is a high affinity biotin which has binded to streptavidin. So I just click on it, automatically it downloads from the internet, and it tries to see, and it also comes up with this particular uh, representation. Now, before moving around, let me explain to you how to manage the visualizer. So in order to zoom in, zoom out, you can use your mouse scroll wheel. But if you're on your laptop, if it supports multi-touch pad, then you can use your two fingers, or in some laptop, it's on the side. You have the scroll wheel where you can zoom in and zoom out. And in the mouse, you can click the right button in your mouse, on your mouse, to rotate and drag it around. And if you wanted to select something, press the left key of your mouse, that is to select. If you want to drag it out, you can press the scroll wheel and then you can drag it out. The same way, you can press the in the yeah, in the Mac also you can press the command line command key in, and then press your right hand mouse key or button in order to move around, rather than the scroll wheel when you're pressing it. Yeah, so these all are the options which you can do it. So to recap, mainly we will be using the right button and the left button of the mouse to select something. In order to zoom, scroll wheels are being used. Okay. And now if you want to change the visualization, you can always go to this particular button, appearance. And then if you want to change the background color, you can change the background color. In Windows, it might come a bit different. Or you can select cyan if you want, you can, it will change. Or if you want white, You can change it to white, right? So you can change the background color. If you want to change the layout, that also can be changed. And if you want a dark environment or a light environment, that also could be changed. If the label size to be larger, you can do that. And if someone is having like color blindness, it will be automatically taking care of green, red, and blue, red. So that will be taken care automatically if you enable it. So that is all with the visualization part. At the bottom, you have something called a sequence viewer. So let me show you that. I hope you are able to see it. Yeah. So on the first, you have the individual proteins. When you mouse over it, you need not to click. You just mouse over it. Everything will turn to be like this particular color. It could be magenta or whatever you call. So it's a typical color will come when you're trying to select it. Okay. And when you're going to residues, you will be able to choose which is the residue. You can see when I mouse overing it, you can see all the residues that is getting highlighted. And coming to this one, it just highlights the ligand. As you can see, it is highlighting the ligand, as you can see here when I mouse over it. And then if any metal atoms, metal ions, then it will also display the metal ions. Water molecules, if there is any water, you can see some water molecules are getting highlighted, right? And then a ribbon, if you want to switch off and switch on the ribbon, that also can be done. This is all for visualization purpose. Now, it got zoomed out somewhere and you want to zoom in, but you want to immediately zoom into the particular binding site. You just press the space bar on your keyboard, it gets zoomed to your particular binding pocket or binding site. Okay, so this is all about the visualization part and other things. Now you can load more than one protein and superimpose them and compare them. Those things can be done, but I will show that in a moment. If you want to select an amino acid, you can click the right button, say hide it or focus to the view. So click on that focus to that viewpoint, it will go to that particular amino acid. If I'm saying I want to go to leucine 39, I'll click leucine 39 and I'll say focus the viewpoint on that. So it will automatically move forward. Now I hope you understand why you require the latest drivers for the display, because a lot of visual importance being given here, because that is what we are looking for, the result interpretation, which will come soon into that. Do you have any questions so far so that I can go to the next? Yeah, nothing much complex, I'm sure. Okay. And I'm again repeating, please don't try to do it when I'm doing it. Please listen carefully. Now you have to be very, very attentive. Let's imagine that you have a protein with a co-crystal ligand. When you have a co-crystal ligand with you, and if you understand that co-crystal ligand's amino acids are your key amino acids, you can select that particular co-crystal ligand. Here in my case, it's biotin. Yeah. And I'm defining this as my ligand as a reference ligand. I can click the green button, which says it is being accepted as a reference ligand. Automatically, software will identify what are the interacting atoms or amino acids surrounding to that particular biotin compound. Now, when I click on the biotin compound, you can see automatically software has generated the binding affinity for the co-crystal ligand. It did not do any docking. It just calculated the binding affinity of the crystallized form. And at the bottom, it will also show you the 2D structure, right? And then here it also shows you the coronas, which I explained to you about the green colored ones. Correct? So yesterday, Dr. Hassan was explaining through using PyMol, and InstaDoc was able to do all the calculation at the end. The complex was also being visualized. So similarly here, we have an inbuilt visualizer, which helps you to start preparing the data. Now, if you want to rename the name, all those things can be done here. And here you can see the binding affinity is coming within a range. Let me explain that very specifically here. Let's imagine Autodoc is predicting an inhibition constant and it is telling 123 nanomolar, 23 nanomolar, or 143 micromolar. None of the biologists can accept that. How can a software predict this micromolar, nanomolar specifically to a value? So that is the reason where we have always challenged when we are trying to transform our docking data to bioassay data. Docking cannot give you a definite value that is 10 nanomolar, 2 nanomolar. It is always can give you a range. Even in dynamics also, when you do the energy also, after several frames, we get to know the range. At several frames, the energy levels are different. And we do clustering and the principal component analysis. And finally, we come up with the final binding energy, considering many factors. So similarly, here we are doing something similar to MMPBSA, and we are trying to calculate the estimated affinity. That's why it is called estimated affinity. Here it is saying within the range of nanomolar and micromolar. But if you say that no, sir, I will publish only with numbers. Yes, you can save this as an Excel file. There the numbers are given. But literally, do you think that these numbers are going to reflect in your biological assays? Never ever. It might be within that range. That is for sure. So that is exactly what we are looking at, right? Now, anyway, this is for the co-crystal ligand. Now here you have the protein separated and your ligand being separated by the tool automatically. Now the next step is that you're still on the protein. Now you can go to protein and say, I wanted to go to protein editor mode. I want to just make some modifications if required. I'm not going to do it, but again, just to show you. I selected the protein, right? And I have several options in the top. If I want to add any atom, if I want to change an element, if I want to change bond type, if I want to rotate or flip, which I told you earlier, if I want to rotate or flip, or if I want to find similar binding sites. And this is where we have the conservation studies. We have to, so if you want to study the similar binding sites of this PDB. So if I click that, again, it goes online. There's a server called as Protein Plus, again, from the same group where they have developed FlexX and other programs. Again, you have to set all the proxy, everything. Your information or your compound information, nothing is sent online. So it uses an algorithm called a Siena, SIENA. And if I click on search, it will send all the binding site information which we have identified from the co-crystal ligand to the server. And after that, it sends back all the PDB IDs showing that which are the other proteins having the same or similar binding site. This helps you to understand about multi-target, off-target, selectivity, specificity studies. So this is something that we have to look at in docking studies. Most of the time it misleads with false positives. So we wanted to make sure we explore all the similar binding sites. Yesterday you learned about a tool called DALI, VASTA, which talks about comparison between two protein structures. Here we are trying to compare the binding site of this protein with the whole PDB data set and then getting back the similar ones. Similarly doing similarity structure, chemical structure similarity, but this is mostly on the PDB. Now you can see these are the other proteins where we have somewhere it is similar to this binding pocket, right? And it gets superimposed automatically. Wherever you see green color, they are whatever I have selected. Wherever you see the blue color, they are the ones which is I have loaded the protein 1STP. So you can see the change in colors in the chains also. Let me go to HSL. Yeah. Here you can see the change in colors. So I'm not going deeper. You have to label them. So in order to label, you can go to label, show label, and then you can click on and then it will show the amino acids, which are these amino acids. So you will be, you can see chain D, chain B. So you will be able to identify that. It takes time for you to get acquainted with the visualizer and other things. But this will be really helpful for you to learn about the conservation of this particular protein before publishing the data. And it can also do similar structure. So biotin was also being bound with certain other proteins or mini protein MP2, M9A, V10A, and also MP2 complex with the streptavidin. So you will be able to identify this. This is really very helpful when you try to do natural compounds or the extracts that you're working on. If someone else has done a paper on that similar compounds, you would load them and try to see what are the other natural compounds or any other co-crystals already being reported on that. Or you have to go for network pharmacology or HitPick server to identify reverse docking mechanism. For example, based on your structure, it will do the similarity with the co-crystal and try to identify your protein structure. That way can be also done. Let me see if any question is there. What is the yellow and red structure, sir? These are the chains. Okay. So the chain is telling you two different. So let me go to the protein just so that you will be able to understand. So this is your actual protein, which we loaded. So the actual protein is in green color now, right? And the protein that I selected 1DF8. This is 1DF8. Anyway, it is showing all. It is overlapping. That's why. So 1DF8 is overlapping with your protein also. Let's say let me give a color so that it will be fine. So I will click my protein and say show as reference. Okay. So that will be always shown as a reference. If you want to change the color, you can click on this color palette and I will say I'll make a purple color so that you will identify, right? This is my the protein that I have taken now. Okay. Now when I'm selecting others, you can see that my reference protein is overlapping with the other amino acids, other protein also. Since that chain is exactly overlapping, that's where I identify they have similar binding pockets. Hope now it is very clear to you. But still you have some peptides in between. Instead of biotin, you have some peptides also, mini protein, right? See, so this is what you can understand. What are the other compounds or anything that is explored other than biotin on the same binding site, right? Anyway, so that is about the protein editor. And if you want to make any flip or anything that can be done, again, I'm going back to protein mode. This is how we just clicked on it. So any mode you are, if your structures are there in the name, near to that there is a menu. Click on that menu, you will see the possible options available. If you still want to go back to and see the similar binding set, go to protein editor, all the data will be still there. Okay. So each modes are of different categories. Any questions? Sir, if we have only ligand and I want to know multiple proteins to which it can bind, how to do it, sir? Yes, Prashant Kumar, there are two ways. If you have only ligand, for example, you have a natural product and you don't know which could be the target. There are two ways to do it. One is you can search hit pick server or SCA. Oh, let me show you from the browser itself. Yes, what it tries to do is it is a hit identification. It tries to do a similarity study between your structure and the structures in the co-crystal of the PDB, and then they try to identify the PDBs there, if it is a PDB. But if it is a target, of course, it is based upon language processing methods. The other one is SCA, ensemble similarity, yeah, similarity ensemble approach. This is also something very similar to, let me find out the link. Usually it should come in the beginning. Ah, yeah, it's here. So this is another online tool which also helps you from your ligand molecule, how to get your targets. Even in StarDrop also we have this feature where you can give the structure from the PDB, it will do similarity search and get you the hits. Another possible way is that stitch. where we do network pharmacology. Stitch database. I'll be soon doing a session on Stitch for the network pharmacology using Cytoscape software. It's a free software, Cytoscape. So Stitch is again a chemical protein interaction network. So here also you can give the smiles of your structure, and then if you are specifically looking at Homo sapiens, you can give that option. Otherwise, you can give auto detect. Otherwise, you can click on Homo sapiens. You're just giving where is it? Chemical structures. You're just giving the smiles. That's all. So you're just giving the smiles of your structure. You search it with a specific species or auto detect. Or if you want to load a mol file, you can click on browse and take it. Okay. So these are the different options you have when you have a chemical structure to find out the protein. These are all online servers. Great. And once you find out, here what is in the CSAR is that to identify similar binding pockets, right? It is based upon the pocket, not on the ligand. So that's why I didn't show you here. Those are the tools based on the ligand. Here it is based upon the binding pocket, right? So conservation studies can be done here. And one more important thing is that these proteins also you have to do docking and see. That's the beauty here. So to which protein it is very selective. Here you see the chains were mostly overlapping, but there are chances where some of the amino acids could have been mutated also, right? If it is mutated, then we have to see mutated if it is interacting properly, non-mutated if it is interacting properly. So those kind of studies has to be done. So docking means not for learning purpose, it's absolutely fine. Taking a molecule, protein and dock and then done, finish. But when you're going for publication, have a depth understanding and knowledge about it. Okay, so we're done with the protein part. Now, next part is that we wanted to do binding site prediction. Keep in mind, I will be doing two methodology. I'll go for the first one. Consider that you have a co-crystal ligand already in your protein. In that case, after you define your ligand and all, again, you click on the menu and say add to binding site mode. You click on add to binding site mode. Okay. I again repeat to everybody, please listen here. Do not do it yourself. You will have 12 days for you to do yourself. Otherwise, you will miss something when I'm telling you. I mean, later if you're doing it. So here we have two approaches to do it. One approach is when you have a co-crystal ligand, the software automatically identifies your residues here, as you can see at the bottom. Here, automatically the residues are identified. You have a blue color and you also have a magenta color where it is saying these are the selected amino acids. Okay. So if I click on this menu again, add surrounding residue, that means these are the amino acids surrounding the particular co-crystal ligand. On a normal range, it is taken between 7 to 10 Armstrong radius. The binding site is calculated. Now, again, let me tell you a trick here. When you use visualizers, there is an option to look at the interactions possible, because if you have done docking in a one tool and if you are trying to generate the interactions using another tool, they always ask you what is the radius or the range they want to collect the contacts. Contacts means the atoms within the amino acids. When you increase it, you will see more interactions. When you reduce it, you will see less interaction. So that is where we are a bit biased and you have to make a understanding. It is completely based upon depending upon your flexibility of your molecule as well as the shape of the pocket. So 6 to 7 or 7 to 10, max is 10, nothing beyond that. 10 is very much extreme, but 6 to 7 is more optimal, or 5 is again, 5 to 7 is more optimal. That is being taken care automatically. So you make sure that when you're making visualizations and all, you take care of that so that unnecessary interactions won't come in. Right? Now, let's imagine when I read a paper, I understand that isoleucine 30. Let me show you. Okay, it's on the other side. It got zoomed up because I pressed the space bar. Yeah, isoleucine here. Just for an example, I'm doing it. Okay. I read that isoleucine is also part of the pocket. If I want to include isoleucine also part of my pocket binding site, I can click the right button on the isoleucine and say add residue to binding site. When I click on add residue to binding site, you can see um, a surface is being defined there also, right? Surface is defined here also. If I say isoleucine is not, then I can remove the residue from the binding site. The surface is being removed. Now here you can see threonine. Where is threonine? I don't know where is threonine now. So in order to choose that, click the right button and say focus view on the component. So here is the threonine. So here is the threonine. Let me see again. Yeah. If you want to label it and see, go to the label, show label and click on it, right? Threonine. So those visualizations will be available. I published in 2016 or 15 in the Journal of Pharmaceutical, American Journal of Pharmacy, where we did a work, and the binding pocket was nowhere reported other than the second crystal paper, and software was not able to identify the pocket. So the pocket was there, but the key amino acids are somewhere closer to that pocket. We had to manually include them. So this is where you can do it. Even in also other methods also you can do it, where you have in Autodoc also you can include those grid to cover those amino acids. All those things can be also done. So you have to make sure that whether those key amino acids that you have selected is the right one, and which not to be required has to be also deselected. Now let's do another method. You have a co-crystal ligand here. Now let's imagine you do not have a co-crystal ligand. In order to reset, you can click on this. You do not have a co-crystal ligand. When you don't have a co-crystal ligand, so let me show you just a minute. Just sorry about that. So the next part is that let's say you don't have a co-crystal ligand and then you wanted to define the pocket. So there you have an option here to predict the pocket, binding pocket. So there they uses doc site server, doc site server. It's a published algorithm. So when you click on it, it will try to identify all the possible pockets. Here you have only one pocket. If you click on this pocket itself, I'll switch off the label. Yeah, if I click on this pocket itself, it will identify the residues neighboring to it. Here, since it is only one pocket, automatically biotin is there. So no doubt about it. But there are certain proteins where you have multiple pockets also coming. So there you have to make sure that which is a key amino acid is in which pocket. And you have to choose the right pocket. Here also you have the same option. If you click on this, you will be able to see add surrounding residues, and you can add or delete. Those options can be done. If you made a mistake, you can always reset it by clicking this button. You reset it. And again, I'm going back to my co-crystal ligand and saying add surrounding residues, right? So these are the options that is available for you to define the binding site. Okay. Now, once you have defined the binding site, you have to click on this button to confirm it. But before that, let me see if there's any questions. Thank you. If you are able to understand, I'm so happy. At this link that shows you. Oh, okay. I'm sorry. That's something else. Great. So now I just click on the green button, which says it gets applied. And when I click on it, now your Now you're, just a minute, sorry.
Dr. Pillai, we are not able to hear to
Oh,
you.
it got muted. Okay, fine. So can you hear me now?
Yes, yes.
Okay, thank you. So yeah, so we have completed that part now. Now, first thing what we have to do when you have a co-crystal ligand is that we have to redock it and confirm whether we are getting lesser RMSD values. Before that, I forgot to mention something. See, you can see here some red balls here, right? All these red balls are the water molecules that is selected. When you're doing the preparation of the protein, automatically the software selected the key water molecules within the pocket. There might be water interaction that could happen, but we have to see after docking. So in order to redock and see whether it is having a lower RMSD or the positions are all proper as per the co-crystal, whether our parameterization is good or bad, we have to do a docking. So I know that many of I have seen, they just simply predict the binding pocket based upon the co-crystal, and then they start doing docking for their compounds without redocking the co-crystal. So you have to ensure that you redocked it, include it in your paper also, that redocked the overlapping, so that your parameterization is checked. See, for your wet lab experiments, you always do calibration. You always use positive and negative control. Similarly, software also require that, right? Your systems are very much dynamic. Any software you do, you have to redock and confirm that the confirmations can be recreated. So click on this menu again, and you will say now you're going to docking mode. You click on this, your structure is being added to docking mode. Right now, you will be able to see your pocket, everything very nicely. For that, in order to enable your pocket, go to this visualization tab at the end corner, and then you can click on binding site. When you click on binding site, a pocket surface will get generated. This is good enough for you to just emphasize a highlight. But if you want to have a little bit more information, you can click on lipophilicity to understand the hydrophobic conditions, or you can based upon ligand element. So you want to know where is nitrogen, where is oxygen, you can also. And if you want to have a clipping surface, for example, sometimes if it is the cleft is very deep, and it is a little bit turnaround, it is not straightforward, you want to see the interactions happening, so you can enable the clip surface so that you can go in, penetrate into the pocket and visualize it properly. So for the time being, let it be here, then I will go move forward. So I just zoomed it to my pocket, and you can see very nicely the pocket where it is being well defined, right? The pictures can be exported, all those things can be done, which I will tell you a bit later. Now, the first thing is that I want to do is I want to do a template docking. I told you there are two docking, template docking and a normal docking. Template docking means it always keeps your reference molecule, whichever you are selecting. If you don't select a compound, then that is not a template docking. If you select a compound, it is a template docking. Two ways it is useful. When you have a co-crystal ligand, it's always recommended to use a template docking so that it takes care of the RMSD, and also it will not deviate too much of conformation apart from your co-crystal ligand. The second study is that, let's say you did the docking already using XYZ software. You want to bring in here, redo the docking and calculate binding affinity and other studies, so that you can calculate the affinity range and everything. You can bring it here and again do a template docking so that it will not much deviate from the confirmation. It will still do confirmation analysis, but on an optimized way. There are several papers where they have used autodock. as well as Glide, and also other docking programs, even Gold, I think. And they brought, after the docking screening, they brought in to CSAR to calculate binding affinity and atomic contribution, and they published it. They are in Journal of Medicinal Chemistry, EJMC, and very well established journals. They have published it. So there is a crisscross, and you can validate it accordingly. Now we want to dock and see, right? So after selecting it, if you want to choose how many poses to be generated, there is a rule here. It's common everywhere. When you have too much of flexible bonds, rotatable bonds in your ligand, there will be so many torsions will be there, so that conformations can be generated. So you have to be very sure how much poses can be generated. Second, if your most of your natural compounds are too rigid, if it is too rigid, by simply giving too much number of poses doesn't make any sense, because it is not flexible, it is not rotatable. So when you are choosing the number of poses, make sure that what are your set of compounds, whether they are too much rings and rigid, whether they are flexible, all those things has to be taken care. Okay, and this is very, very important that you have to check it out. Let me also look at something. Okay. Any doubts? Let me check the chat if there are any doubts. No? Clear. And clash tolerance is also very important. I told you interclash or intraclash is also something important. So that also has to be taken care. So if you give a very high tolerance, then it will be balanced, and you have to check how far the clash tolerance taken care. Otherwise, I would recommend initially to be keeping it as a standard, right? And this one's example, I will tell you a bit later, because here we are trying to define the docking definition. We are trying to export it. So when we are exporting it, we can use that in a Flexus command line for the virtual screening. So this I will explain it a bit later. Now we will, after defining the poses, we are just doing a template molecular docking. So click on it. It is going to do docking, and the docking is done. So that's why I said it will be very fast. My machine is i7. Yes, that's could be the reason why it was fast, but normally it is fast actually. Now it has done different docking. Now let me do something. If you go here, you can click this button and say show as a reference. So your co-crystal ligand will show as reference, and I'll color this as probably a red color so that it will highlight. Yes, this is my co-crystal. Now I wanted to click on my docked. Yeah, I got the first few of them exactly same. Some are slightly shifted. You can see the difference now, right? This is exactly what we are trying to do to see by redocking whether we are able to recreate them. But still you can see there's a tilt and some conformational changes, right? So this is only the docked poses. Now you might ask me, so where is the score? I didn't do virtual screening, so why should I need a score here? Now then I select all the poses. I ask them to calculate the estimated affinity. So click on estimated affinity. Now it will do hydrogen bond network, height calculations, torsions, clash properties, everything will be taken care. And then the color code, which I have told you, will come in, where there will be more interpretations could be possible, right? So let me do that, just a minute. So any questions until it gets finished? No questions? Do you have any questions before we proceed to next?
I guess no.
Okay, great. So we'll go further. Now, we need more information to confirm whether this pocket definitions are all fine, right? I will look at the visualization later, but I have some more information here. So I have calculated my estimated affinity. I've also calculated lipophilic ligand efficiency Okay. And then I will also calculate probably the clashes, right? And also torsions. So this is all trying to tell me whether this is a bioactive conformation or not. And then if I want to sort, which is the best affinity, I click on this, it gets sorted by themselves. You can see most of the pocket is almost similar. It's not deviating much. And you can see the nanomolar micromolar range. But let's see what is a co-crystal's binding affinity. So co-crystal is here. The top table is a co-crystal. Below table is all docked portions or generated portions. So if I click here and then I will say calculate estimate affinity. So it will calculate the estimated affinity of the co-crystal ligand, not the dot. Right? So here you can see it is within the range of nanomolar and micromolar. When I docked it, I also got the similar nanomolar and micromolar range. Here I can see the ligand efficiency is good. That is why thumbs up. And all the green color is showing me the intra and interclash. So let us visualize now what is all these colors and representations means. Okay, so for that I again go to enable torsion coloring and clashes. And I will change this color probably, no, I will take it off as not to show as a reference. Yeah, now I have the color codes. Let me label it by going to label and measure, click label. If I click on this particular torsion, I can see that this particular torsion is in the presence where 55 degree, yeah, that is the minus 55 degree is the torsion here. Or 56 it's given here, yeah. And about 55% of the compounds in the Cambridge structural database has this particular torsion. So this is crystallizable. So that is what we are looking at. So we are trying to identify whether these torsions are crystallizable or not. Now let us look at this particular bond. Here you can see the torsion is minus 87 degree. And here it's only around 3% is present in the Cambridge structure database. Now here you have a green colored one, right? That particular, let me zoom out. And here you can see the torsion is minus 179 degree. And this is almost 93% present in the Cambridge structural crystal structure database. So this conformation, bioactive conformation, is a crystallizable conformation. That's what we come to a conclusion, right? Based on the torsions. Now, let's see some other, because many of the publications, they post this picture to show that, right? Now, let us also look at these coronas. When I click on this particular carbon of the biotin, I can see the hide energy is minus 5.3 kilojoules per mole. What it is trying to do is that we are trying to understand this particular atom is a contributing minus 5.3 kilojoules to the total binding energy. So keep in mind, I'm not talking about the molecular binding energy. I'm talking about the total binding energy. If I click on this particular atom, even though it is contributing, but it is very less contributing, it is of minus 0.6 kilojoules per mole. And this is what on the desolvated state and the solvated state, they are being calculated and they are being summed up to give the total height. So this is called atomic contribution to the binding affinity. If I go into this particular nitrogen, you can see it is minus 4.2 kilojoules per mole. And as well as for this nitrogen, here also it is minus 4.2 kilojoules per mole. Now you might think, sir, is this going to change as the conformation is going to change? Yes, it is going to change. Let me choose a conformation which is here, which is having nanomolar and micromolar. So for whatever displays that we are doing, let us do for this one, it's slightly different. So let us see how far the displays are there. Now for this nitrogen, earlier it was a green color. Now it is red color because the hide binding affinity energy has gone to negative contribution with minus 5.5. And here the nitrogen, earlier it was not torsion, sorry. Earlier, this nitrogen was having minus 4.2 or something, but now you can see the contribution is minus 2.0, right? So this is why I said not based on score, you choose the final component. It's completely based upon the contribution. And here, again, earlier it was around minus 7, I think. Now it is minus 5. So for each conformations or for each poses, the contribution of the energy from each atoms will be different. Now, if you want to see the interactions that is taking place, we can enable the clipping pane like this, and then the clipping pane, and you will be able to see these are the interactions happening. So there is an interaction happening with water molecule also. You can see here some interactions on the water molecule, and now you can see, let me zoom in. Yeah, with this oxygen, there is some interactions happening. Let's see which are the amino acids. This is ASN23 in Cheney, and this is serine 27, and which is this one, tyrosine 43. So these all can be defined here itself. Now, if you want to make it look nicer and other things, always you can play around with your, the residues, or if you want to take away the chain, you can take away the chain. And then I zoom out, and then I will take off this and also the clipping panes, and then I can display it with the interactions. Or if you want to change the colors, you can change the color. Those things can be done here, right? So it's up to you. So you can use different visual, even PyMol if you want to make it, you can use PyMol. So it depends upon how you, but all these information you cannot put it in those visualizer. So this visualizer is specifically for showing the torsions and everything which has come in. So those information you might not get with other visualizers. So it's up to your personal choice where you want to, how you want to showcase. Many of the publications, you will be able to see the picture like this, where they show both the pocket uh, as well as other informations on the torsion. Now, if you say, sir, no, I want to see the values. I want, without seeing values, I cannot do it. If you're in that particular way, then you can always go select which are the molecules you want to export. You have a button here, save button, click the save button, and you can click, you can either save it in SDF format, or you can save it in XLS file, right? It is Excel file. So that when you save it as Excel file, you will be able to get the values, the minimum and the maximum values. Okay, any questions? Oh. Oh, no, something different. Okay, so no questions so far. So if you want to export, now we did redocking, everything is fine. I will check, revert all, uncheck all, and I will check only this one, the first one, because I want to keep that, retain it. And then I will say when you're selecting this invert selected, so others will be selected, I will delete it because I don't want others. I want to just keep one for reference. Right. Now I want to bring in my molecules in order to dock. For that, I told you already, you can go to analyzer, right? And you can create the structure. Now let's imagine you want to bring in a structure. Okay, let's go to PubChem. And I will take a small molecule. I'll take the smile. I just copied the smiles. Okay, that's it. I just copied the smiles. I came here, just pasted the smiles. That's all. 2D structure and a 3D structure. Of course, it will be outside the pocket. Don't worry, we have to dock in order to get inside the pocket. So you can copy paste the smiles. As I told you already, the software has an engine which takes care of 2D to 3D optimization, including ring conformations. Everything will be taken care of. Now let's look at some other similar compounds. Or maybe a little bit lengthier. Yeah, let's try this one. The smiles will be at the bottom, scrolling down. Yes, it's here, canonical smiles. And paste it here. And when I click on it, that's a structure. Now, only drawback is that when you copy paste the smiles, of course, you are not giving the name here. So you have to double click on the name and you give the name. I'll say demo 1, demo 2. You can do that. This is one way to do it. The other way is that you can go to this file menu, load molecules, and you can load the molecules, right? That is the second way that you can do. So I have some molecules already here. So let me pick them up. So these are 2D. So automatically the software is converting 2D to 3D. And then I have some sample molecules here, right? I think I worked on some project and those are the molecules here. So these are all here. Now I don't want all to be added. So I want only some compounds to be in my system, like to dock. So I will select this molecule, this one, maybe camptothecin and xanthine. And then here there is a button called as where you want to add or move these molecules to which mode. Click this icon and I'm saying add to docking mode. When I click on add to document, whatever molecules I have selected, that only comes to the docking library. Okay. And I already said that biotin has to be my reference molecule to dock. But let's imagine I don't want to do a template. You don't have a co-crystal ligand, but you have a pocket already defined. So you want to see docking with all these molecules, what will happen? This I will define it as co-crystal dock. So I can rename everything in the table. Right now I'm going to do docking. So here also again I'm keeping it as default 10 values. If you want, you can increase up to 500. I'm not doing that. It will take time. And then I will generate poses. So when I click on generate poses, all these compounds, each of these compounds, it will do 10 different pose generation. Let it do it. But do you have any questions?
There was one question from Gagandeep Singh few minutes ago.
Okay. Okay.
Let me see if it is in the chat now. Yeah, it was which tool do you recommend to fix for missing residues and loops in the model or X-ray crystal structure as per your experience?
Modeler, the best one.
Okay.
Modeler and Rosetta is, that's what you have to also understand. Every year they conduct competitions for these software companies and algorithms to understand which one is predicting accurately in par with experimental data. So when you check every year, the protein modeling, Rosetta is the most accurate one. The second is Modeler. All the paid softwares are below that only. So for me, from my personal experience, Modeler and Rosetta is the best. Rosetta is a little bit, learning curve is there. So the easiest one you can learn is Modeler. So Modeler is anyway free for academics, so you can do that. And second, all the loop modeling, refinement, everything can be done in Modeler. And when you, after the modeling, you have to again do dynamics in order to understand whether your backbone is stable or not, to minimize latency. So that is a tool I recommend. And I have already shared my links in the GitHub. So if you still don't know how to run Modeler and other things, you can use the Jupyter Notebook, which I have already shared the links earlier in the morning session.
Ms. Savanu, do you want to ask something? You are unmuting again and again. Please continue, sir.
And you can see here, CSAR is taking 213% of my CPU. That means it is utilizing 3 CPUs at the same time. So CSAR automatically paralyze from your computer based on the availability. You need not to mention. But let's say you are going to run CSAR on a 16 core or a 8 core machine. You don't want the whole thing to be used. By then you have to want to limit the usage of the number of cores by the CSAR. Then you can say CSAR that to use only 4 core or 8 core. The advantage there is if you're giving 8 core, at the same time, CSAR will run docking for 8 compounds. So if you're doing a virtual screening, at the same time, 8 compounds, so that your calculations will be much faster, not like one by one. So here, the calculations are much parallel. So that's why I said it is faster when compared to others. Right? If you have only 2 core, of course, it will run on 1 core because all 1 core may be used for others. But while the job is running, ensure you don't disturb it. And also keep in mind, don't use Chrome browser, all this Opera browser, Edge browser, while any calculation, not only this software, any software you're running, please don't use those browsers because those browsers take a lot of RAM and CPUs because you're using multiple tabs. So the calculation is over, right? Now let's go back. I don't see any questions on the chat. Now it has been docked, but it is not showing any affinity. So only the docking has been done. As I told you earlier, if you want to get the score for virtual screen, I didn't do virtual screen. This is not at all. I cannot call it as a virtual screen. So when I do more than 500 or 1000 molecules, of course, you can use FlexX to get the score. There you get the score. So now I selected all the molecules and I'm asking it to calculate the affinity. So estimated affinity. It will also take slightly time because it has to do solvation, desolvation terms, and then it has to calculate the atomic contribution. So it will take slightly some more time. Okay, there is another question from Gagandeep. Can we compare two poses of protein ligand complex, suppose initial and final frame with water and ions removed after MD simulation to visualize RMSD and pocket changes, if any? See, CSAR does not do any molecular dynamics. That is a confusion here with everybody because when you are using packages like all-in-one means you have everything in one. You might get confused on which tool uses what. And because workflows, when you click on a workflow, multiple packages comes into picture and does the job with you. But here it is dedicated only for docking. So if you want to do anything with molecular dynamics, you can use it with the Desmond. Desmond is free of cost for academics with the GUI. You can use GROMAX, you can use NAMD or even Amber tools, which is again free for academics. Amber tools I'm talking about, right? So the question here is, can we compare two poses of protein ligand complexes and final frame with water after MD simulation to visualize and pocket if pocket changes in CSAR? You can visualize the complex. Yes, you have to make a complex as a PDB, bring it back to CSAR, and again you get it to analyzer and try to calculate the binding affinity. So whatever the MD frames that you brought in, it will not change. The same thing can be retained and binding affinity can be calculated to see, compare and see MD simulation affinity and this one's affinity. Right, and just like you mentioned about structure comparison earlier by importing through CNF, right? But to compare to, yeah, that's what I answered your question, Gagan. I think you understood now. So you can bring in the frames. Of course, it is a complex PDB you have to bring in. The file format should be PDB. So with the protein as well as with the ligand, you can bring in. And then you can predict the atomic contribution and then predict it. That is possible, yes. So now I am just doing it. See, I can see co-crystal is still the best based on the affinity. And the next I can see, let me see which one is. The next I can see the demo 2, which I have taken from the PubChem. And then the xanthan. But xanthan is in the range of micromolar to millimolar. But demo to this particular compound is in the range of micromolar and slightly into nanomolar, but still our co-crystal ligand is the best, right? Now let's look at a bit more detail about the demo 2. Yeah, so here it's all favorable. Only torsions you can see they're a little bit having issues. So we have to look at different torsions, or probably it is more rotatable bonds. That could be also an issue. You can increase the poses for this. Here you see the torsions are absolutely fine, but your height is bad here. So let me look at the height of this carbon. Oh, you can see here 9.1 kilojoules per mole. It is negatively contributing to the binding affinity. That is the reason why it is in micromolar. Now let us see this demo 2. So demo 2, this one is highly contributing to the binding affinity, which is minus 7.5. That's why it is still in the nanomolar region, within the nanomolar region, right?
Is there a way
And
to remove these torsional stresses?
as I told you, you can increase the number of poses from 10 to 32 or 40 maximum. For small molecules, it is 40 is optimal, so that it will try to generate all possible conformations. 10 is anywhere restricted, right? So if you make it 40, it will take more rotations, and then it will try to see whether it is possible to avoid it or not. Because when you ask me this question, you also have to keep in mind it is trying to fit in based upon the whatever it can fit in. We cannot forcefully fit in, right? So it will see its best what it can do. The other forceful fit in is something that is pharmacophore based. This one, defined pharmacophore. That is also an interesting, very interesting tool. For example, in from literatures, you understood that this particular protein is, or this region is hydrophobic or hydrophilic, and you want to have some interactions there. So when you do a big virtual screening, like 5 lakh compounds you did, you based upon the score, you brought in 1000 molecules. Among these 1000 molecules, I told you in the morning, you have to look at whether it is interacting with key amino acids or not, all those things, right? But if you want to make your life much easier, you can define pharmacophore constraints on the receptor. So when I click on this, you can define new constraints, and you can say that whether this particular oxygen, I have an oxygen there, okay? So let me enable, yeah, or clipping so that it will be easy for me, no? I'll take this off, yes. So I'm just adjusting it to see if I can fit in properly. Let me check. Just a minute. Yeah, create. I'll click on the since I'm sharing the screen, it's a bit irresponsive. So let me check again. No. I'm having difficulties. Sorry, I have to stop the share and then share it again, okay? Because it is, let me, yeah, now it is moving when I'm stopping the share. So let me do it once again and see if it's working fine.
खालू कहाँ देखें? खालू में कौन सा?
No. Can you see my screen?
Yes, sir. Your screen is not visible, sir.
Oh, okay. So let me share it again. Just a minute, it's taking time. Okay, I don't know why it is so slow. Let me try again. Can you see now?
Yes, sir, it is visible now.
Okay, great. So let's look at this, and then also the element coloring. And then I will define the pharmacophore, and then yeah, now it is moving. So I am dragging and putting it to the oxygen verify. So you have to rotate it in all possible ways to fit in it properly. Okay. Yeah, now I am actually covering it properly. Now you can say whether I need to have any nitrogen or oxygen in the ligand, or whether I should have any halogens or donors, or it should be a hydrophobic, you can mention it. Or it should be any carbon or any nitrogen or any non-hydrogens or any aromatic you can define. So this is very helpful when you're doing virtual screening, because we don't want to sit and look at everything that is whether it is interacting with that amino acid or not. So you define this and then you click apply. Then you say this will be active. If you say this is going to be very strict, without this interaction, I don't want any compound in my list, then you don't check this. But if you think that it's okay if I have interaction with that amino acid or without that atom, is it fine with me? Then click on optional. You can add a new constraint again. You can either include or exclude. So exclude means I don't want an interaction with this particular amino acid, right? That is what you're looking for. So this is all based upon literature data only. You will be able to confirm whether this is required or that is not required and all. So you will be defining it. And then you will say, I don't want any of the aromatic rings there, because if your cleft is so small and if the aromatic cannot enter inside, then what is the point of it, right? Even though it will give you a score and it may not interact with that. So why you want it? Then we say apply and say, I want this is to be very strict. I don't want to, I want to exclude this. And you can give what is the affinity, I mean coverage of the radii. The radii can be changed here. So before defining, you can increase and decrease and increase the radii here. So this is again, you're bringing in a little bit of biochemical aspect, saying that this is very selective. Without that, I don't want to do any docking. And if you want to delete this, click this and say delete constraint. And again, click this and say delete constraint. That can be done. So that is the importance of pharmacophore. And this also very much important when you're trying to do scaffold hopping. So when you define it and when you do fragment-based design, so you need a specific interaction in that particular atom. So it will search the database, which could confine with whatever constraints you have given in the pharmacophore. That can be also done. Any, sorry, I didn't check the messages. I don't know if there's any questions. Let me check. Okay. Okay, great. I don't see any questions here. Before I move forward to fragment based, do you have any other questions? Vandana madam.
How do you do virtual screening of thousands of compounds here?
Yes. So again, you can go to analyzer or directly from the doc itself. You can go to see this menu, go to load molecules, load SDF or mol2 files. We have tested up to 50,000 molecules in CSAR. It seamlessly works fine. The loading of molecules will be much faster if it is already a 3D structure. But if it is a 2D structure or smiles, the software will take little more time to load it because it will convert from 2D to 3D. And then only it will load to the software. Now, above 50,000 or 1 lakh molecules, we recommend you to use FlexX command line, because CSAR also uses the FlexX as for the docking, but the problem is there'll be a lot of data to be loaded, right? So if your graphics card, if you don't have a proper graphics engine, maybe less than 2 GB you have, then it might crash. It's not because of the software, it's because it does not have the resources. So when you load large amount of data, it's always recommended to do it in the command line. Otherwise, up to 50,000, it's very stable. We have tested it, or even on a 4 GB laptop. So answer to this question is yes, up to 50, you are fine. Anything above 50, I recommend you to do it in the command line. Now in the command line, which is, I purposefully left this, which I am going to tell you now. Once you bring in the binding pocket prediction and bring it to the docking mode, you have this button called export docking definition. You have to export this. This is called, it is clearly mentioned here. This docking definition is in order to calculate the docking using FlexX. So what you're trying to, your input file for your FlexX is only just two files. One is this docking definition. The second is your input ligand library. It could be thousands, lakhs, millions, billions, no problem. Ensure that you have 3D molecules there. There it will not accept 2D. It will accept only 3D molecules in FlexX. In CSAR, it accepts smiles 2D, 3D. But in FlexX command line, it will accept only 3D structures. So the two input files required. One is this particular file which we are going to export, which has the information of the protein, co-crystal ligand, binding amino acids, the radii, the shape of the pocket, everything is already there. So that's why this information is important. So you can use CSAR to define your pocket, save it as a file, and your SDF 3D format of your ligand. Only these two files with a single command, it will be able to run. Let me try to show you that. Let me increase the size. Give me a second. Okay. Sorry. Yeah. So you have in your Windows also, when you download and install, it's just a single file called FlexX. You can use the same license file which the organizers have shared with you. Just to put the license file in the same folder where FlexX is there, right? So now we are going to do the definition. So the only command, this is the same command for everyone. You just follow. If it is Linux or Mac, it should be FlexX. If it is Windows, it is just flexx.exe. Okay. If it is not Windows, the Mac of it is just FlexX. That is the first thing. Second, if you go through the manual itself, here there is a manual. There also you can give it. But let us give it docking definition. So this is the file that we generated from the CSAR. So I'll give one stp.ecf. Okay. ECF is the file that is generated from whatever file name you are giving to save, that can be saved, and that same file name you give it, and ensure that it is in the same folder of the FlexX. Otherwise, you have to give the full path of the file actually. So input is ready. Your protein, pocket information, reference ligand. If there's no reference ligand also, if the pocket is defined, that is enough. All things are come. If you want to define pharmacophore, that also can be defined. Now you are defining your input. Input means here you have input means it should be 3D coordinates, right? And also it should be SDF or mol2 file supported. So I will give ligand.sdf or mol2. Both will be supported. Now you have to give output. After the docking, your output is also required. So you will give -O, and then your output will be in SDF format only. So I will give output.sdf space. And then I want to see the result, right? I can give -V and then 4. So when you give -V4, let's say you have 1000 compounds, If any compounds are having error or if it is skipping, it will show which compound skipped and all. So you will get a little bit more information about what compounds did not dock and what compounds docked, all those information. This is the only command required you to do, nothing else. Once this SDF is loaded, the output has come. Either you can put it in Mona to visualize your SDF, or Mona is again a free software for academia, which is provided by Biosolve IT, or you can load it into your CSAR in and under analyze. Don't load it in anywhere else. Under analyze, you will be able to see the scores also. Docking scores can be seen in the SDF file. Now let's imagine you have billions of compounds and you have a computer with 32 core or 12 core. Now you can also mention how many cores to be used for this particular calculation. For that, you have to give thread count, and then I will just say 16. Means at the same time, 16 compounds will run on 16 cores. So your calculations will be much faster. Let's say you don't have that much. You're using an i5 with 4 core. You want to make sure that this is going to take time. So I want to use it only on 2, because other 2 I want to use it for some other purpose. You can restrict by giving 2. So the calculations will run only 2 at a time. So this is only the command you're going to do. If you have any doubts on this command, please let me know.
This you are telling for less than 50,000 molecules, or I mean everything, or more than
everything. For example, if you want to do, or if you want to if you're very sure that you want docking score also for your studies, then you can do this even for 10 compounds, 20 compounds, 1 compound also. It's up to you. But highly recommended for large number of compounds. And if you say that by default, the 10 is the poses that will be used, 10 confirmations will be generated. But if you think that you want more, give 10, give 20. So for that, you have to give max number of confirmations is equal to 30. If you want to increase the poses. Okay. So this is the maximum thing that you need to give. Okay. So nothing hard, no coding, no scripting, nothing required. You generate the ECF file from CSAR and you have your ligand molecule in 3D format. Ensure that it is in 3D. You can use OpenBabel to convert 2D to 3D. It's a free tool, and again, and this is the standard command. Ensure that these files are in your folder where Reflexix is there. Otherwise, mention the path. So this, through this, you will get the doc score, not the energy doc score. Okay.
Reflexix is integrated with CSAR, no?
Yes, Flexus is integrated with CSAR, but there you don't get the docking score, because there the CSAR's main objective is to find out the binding affinity and binding energy, not score, because nowadays nobody reports score.
Okay.
Score doesn't have any value apart from virtual screening.
So if you are screening large number of molecules, you can just only rely on the binding affinity. No need to shortlist the molecules based on the scores.
I think there was a confusion. If it is a large number of molecules, you can do virtual screening to calculate docking score. Based on the docking score, filter the compounds, bring it down to CSAR, again to trim down the compounds using binding affinity, energy, and interactions with key amino acids. But if you have only 5 or 10 molecules, you need not to look at the score. Directly, you can go to CSAR and then look at the interactions, binding affinity and all. Because I have seen many papers, even during the submissions also, many people just looked at the score. They didn't even care about the interactions that is happening with the key amino acids. Based on the score, they reported this compound is best. But how it is mechanism wise, it is going to help, not at all reported. So that has to be taken care. So there's another question from Gagan. Don't worry, Gagan. Your job is to ask question. My job is to answer it. So don't worry. So can you suggest any open source tool to calculate MMPBSA of the dock ligand to target during the screening after ADMET calculation and before running MD for final set of ligands? As you mentioned in the drug side is doing the similar to MMPBSA in CSAR. Yes, I understand. I do not know any MMPBSA based tool without doing molecular dynamics. After doing molecular dynamics, of course, you have many tools. Even I have also posted my tutorials, and there are latest versions of MMPBSAs also, different versions recently came up for GROMAX, Amber, as well as for NAMD. So for NAMD through VMD, my tutorials are there on the YouTube where you can calculate the binding energy calculation. But keep in mind, by default, it will do 7 different MD simulation calculations, 3 of them on 200 nanoseconds, not 100 or 50, 200 nanoseconds. Ensure that you have good enough of computer resources to do that. So, but otherwise, I know only HIDE which can do. But there were earlier Sybil was there which was able to do it, but Sybil is end of life. There's no support hereafter for Sybil, so I am not sure. So yeah, without MD, MMPBSA, because most of the MMPBSA codes require the trajectory data and other things, including many series of calculations. So otherwise, I know only HIDE. Yeah, unfortunately, it is paid. Okay, so shall we go to the fragment based?
Yes.
Okay, great. So let me try to pick up a compound which is problematic to do optimization. I will take a demo 2. Yeah, I'll take this compound demo 2. I want to see how I can improve this particular compound, right? So I will take demo 2 and then I will say add to inspirator. So demo 2 or whatever compound you want to optimize. See, when I'm saying inspirator, it is fragment based approach. So the rule number 1, you need to know a chemist who can tell you that this can be synthesized or purchased or anything. So add to inspirator. Now we are in an inspirator mode. When you're coming to inspirator mode, there will be a bulb, light bulb will be there. Ensure that the database is also installed. If your database is not installed, it will not work properly. Now you have the ligand with you. Okay. If you want, you can also look at, if you click on it, it will show the binding affinity contributions and other things for representations. Now, 2 things has to be done. We have to define which region of the compound has to be replaced. If I think that I want to replace this particular region, right? The umbrella is facing towards that. You can see here this is being faded and the other region are highlighted. Now, automatically the algorithm kicks up saying that this is a growing algorithm, right? It automatically replaces that region with another fragment. If you think that I want to replace, and if I want to cancel this, click it again, it will get canceled. I want to replace this particular region, right? I want to replace only the middle region. I don't want to replace anything else. So this will be there and this also will be there. This is called a core replacement. Now let's imagine, let me delete this to show the merging, because I'll go back to the docking. If you want to merge 2 different ligands, let's imagine I want a biotin, that is a co-crystal ligand, as well as I want my demo 2 molecule, right? I want these 2 to be merged. So I will select those 2 and then say, go add this to inspirator. So I have 2 molecules here. It has to be done together. That's why I deleted it and brought it. So I have 2 molecules together. Here, I have to mention which regions to be merged. I will say this region, now this region, yes. This is now I'm just playing around, okay? So you have to really make sense when when you are doing it. And then in this particular protein compound, I want to select this region. So this aromatic ring and this region will not change. The other 2 region will change and merge. So here, if you see this particular 2 ring, you can see this is of this particular structure. That will stay there. And this is the aromatic ring of this structure. and the middle region between these, they will get merged. That is what we are trying to do. Once it is done, this will automatically enable saying that linking and merging. You can do it for 2, you can do it for 3, no problem, not more than 3. And press it. It will search the database, whatever database you have installed, whether Zinc database or PDB database, and the 3D positions of those, whichever we ask not to change will not change, but the other portions will try to find out the fit in. But here the problem is, when we did not give any constraint, it might overlap with your pocket. It might overlap with amino acid proteins and all those atoms, all those things. Let's check it out. So when I click on this ligand, you can see this. This is a new replacement. They merged both biotin and the compound that I taken from PubChem. So this is called merging. You can see some abruptly going out and out, right? We have to dock it and see whether it really works or not. In order to do this much more meaningfully, what we can do is that, let me do that. Yeah. In order to do that very much meaningfully, you can define the pharmacophore constraints. So let me do that once again for you. Just a moment. I have this compound with me. Okay. Sorry. Yeah, I have this compound with me. Okay. No, I'll do this example only so that it will be easier. So with this example, what I want to add is pharmacophore constraints. I'm saying that I don't want, I want to have some acceptors in this region, right? In this particular replacement, I need some acceptor in this region. Without acceptor, don't make any choices. Or I can say that in that region, I need any nitrogen or oxygen. Okay, that I'm going to add. Then I'm also saying that add new constraints, and then I will say exclude maybe a spiral center or any non-hydrogens or maybe aromatic, and then I have to put it slightly outside the pocket to say that nothing has to be replaced here. So I don't want anything in this region. So don't get expanded out. So automatically the software will understand, okay, no fragments has to be generated in that regions if any replacements are happening. Right? So this is all you have to do. As I told you, a chemist only can tell you whether I can make a replacement there or not. First of all, because that two compounds to be broken down and then merged, right? So that's why I clearly tell everybody that chemist is required here. Okay, so I did that. Now I'm going to do this. Let's see what we get here. Oh, is there any other questions? Doesn't it require structure optimization, minimization after merging? Of course, yes, again. After replacement, you have to further do optimization and docking. So that I will show you next. Without that, we cannot confirm whether it is interacting properly or not. So great, we got some compounds. Let's see. See, I have a nitrogen here. I said I need a nitrogen. So there's a nitrogen there. See, wherever I mentioned the spatial, the ring did not come. Very close to that, a ring came. Yeah, I got another nitrogen. Yeah, there is an oxygen. Again, it's not an aromatic ring. Yeah, it's not an aromatic ring. It's a 5-membered ring. So let's take up the deviation, less deviation one. This one is having a very less deviation. So I'm taking this. So you have to select this and say add to docking mode. Right now, this is my compound. I'll say Recore 1. That is my compound. And I will delete all. Maybe, yeah, I'll delete all. Click and delete it. And to make it much more nicer, I will delete these compounds. to make it a much better view. I don't want these, just I want to compare with my, yeah. And then I'm going to do docking, and let's see how the results are coming in. So a lot of care has to be taken here. So now here it takes care of optimization. It gets a 2D, 3D confirmations, and also it takes care of ring confirmation of your newly fragmented composite structure. And then it will do the docking. So answer to Gagan's question. So until unless you do docking, they will not take care of optimization. It will just show you the in the space. It will show you the fragments. Morning there was so many questions. Now there is not much questions. So I am worried that are you able to follow me or not?
No, we are following you.
Maybe when we'll use the software, then probably the
Yes.
questions will come.
Yes. Hopefully you will make a lot of rewinds to the YouTube in order to understand that. Yeah.
Yes.
Okay, and I'll calculate the affinity. Yes. Let's see if there is improved or yeah, it's bad actually. Right? See, it is not better than biotin anyway. Right? So a lot of optimizations are required. I just now played around, right? So a lot of optimizations are required. You have to take much care on improvising things. So it's not simply that you can do. This is again a fragment based approach. But if you're thinking that this compound is having issue, let me do a molecular editing. So you can go to add to molecular edit, right? Here, if you click, you will be able to see, yeah, what are the problems it is facing this atom. So let me once again see, yeah, this nitrogen, right? So I can think of replacing this nitrogen maybe with the oxygen. I can do that. So what I will do is, I want to change the element, right? So for that, first select the element, this one, nitrogen, it is selected. Then I'm going to change the element to oxygen. Click on oxygen, it has changed. Now, after changing, you have to make sure that you also do docking, right? So you do docking, and then it will get added to the table. So this again, you need understanding about organic person, because whether we want to know whether this can be possibly replaced with oxygen or not. Synthetic feasibility, accessibility, everything is taken care, but why to take chances? Always it is better to check with a chemist. So here, when you replace it, optimization, affinity calculation, everything takes care when you click the docking button. Great. Now let's see. Slightly improved, right? So after changing to oxygen, it has slightly improved when compared to the protein, but doesn't look great, right? This is the nitrogen one, and this is the oxygen one. Yeah. So several optimization has to. So if you are doing any optimization, yes, this is how it takes time for you to come in. This is not a one step job and all. And fragment based, of course, it can generate a lot of compounds with better activity. But again, whether it is synthesizable or not, you have to always check it out. So I think I have covered most of the modes here and result analysis part. And if you want to export these compounds, as I told you, you can export it as SDF. You can take it to a different visualizers, load the PDB there. As I told you, PDB is always, the proteins are always rigid. It's not going to change. Load your PDB there and these poses in SDF there, and you can generate if any other visualization required apart from this. So otherwise, I think I'm almost done, but if you have any further questions, I'm happy to answer.
Any questions from the audience? You can unmute yourself and ask.
They are tired.
Very
lengthy
now.
day.
Yeah. Now everybody needs a tea. I guess so.
So if you have any questions, you can always email me, no problem. Okay, another question. So as a microbiology student, this is very new. Sometimes all this at once, yeah, you're right, overwhelming. Can you please give suggestions? How can we start to become familiar with the tools and this field? The best way is, see, many people, including faculties, used to approach our company saying that, can you give training? We say no. Until unless you try, you will never learn. So the best way to learn is take a project of your interest. It could be something related to your current project also, and try to incorporate in silico work there. And through that you will learn, because there is a need for you to do it. So automatically you will learn. It's not about free software or paid software. Let me be very open and frank. I know this is on YouTube. See, don't get handicapped. Handicapped means only learn one software and you always die with it. And if some else, and if when you go some other opportunity, different institution, if you don't have different software, you say, I have to again learn it. When you're a student, that is the best time to learn multiple things. So learn multiple tools which can do docking so that you are not handicapped. So start with a project, and through the project, you learn things. But just for learning tools, don't go anywhere. Waste of time. Because until unless you apply somewhere, you really don't understand how this has to be worked. Now you attended this today's 4-hour sessions. Tomorrow, if you try to do yourself, you'll have 100,000 questions there. How to click this, what it is happening. So you have to again rework on. So multiple times you have to rework and then apply on your project. You will have hiccups. You have to ask questions to people who know, and then that's where you have a better understanding how it can be applied. That's where you transform information to knowledge. Until unless you apply your information, that cannot be transferred to knowledge, right? So you take a project, do it, and then you will learn it properly. Does QSAR consider amino acid residues defined in the binding site to be flexible while docking? I already told you, Gagan, these are all rigid, nothing flexible. When you're bringing in flexibility, small MD force fields, everything comes into the picture. No artifacts here, so it's rigid. If you want, you have to do MD simulation. Like propose a question and see which tool will help in this and then work on it. Not like propose a question. What you can do is, since you are, as you rightly said, you're very new, you can take a paper where again, microbiology studies are being done, as well as some in silico work is being done. Try to recreate it. If they have used paid software, recreate it using free software. Okay, don't worry about which software they have used. That's what I said. Don't get handicapped. So try to recreate first a published paper so that you know what you have to expect for the results. Then apply for your project. Don't propose from the initial way and then you will find difficult. So always recreate or reproduce a published or a already worked out well data, and try to take it from a very good group of people who have done. I'm not referring to journals. a very well established group who has published a work, that you try to reproduce it. Okay. If you are able to reproduce it, that means you learned it. And then now apply it on your project where you propose a question and then see, evaluate and check it out with different tools and then come up with your hypothesis. Yeah, it takes time. You need to have some patience. And just a training is not going to help you. You need to try yourself. So that's the purpose why we have given you 12 days of license. Just giving 4 hours of training, and if you're not able to try, what is the purpose? Now, if whoever is serious, from tomorrow, try to reproduce from the publications. There are a lot of publications on CISA. You try to reproduce some of them. PDB IDs are there. Try to do docking and see whether you're able to get the same result, and then try to work on your data set and see if it works. That's the best way. No problem, Muskan.
Any more questions from the audience?
So I'll be so happy if at least many of you students, not about this software I'm talking about, at least you get into this area which can help your field. I don't say you have to get into bioinformatics, never I will say. Because I don't say because it's bad, no, it's good. It's because you're already doing microbiology, zoology. Whatever project you're doing, try to incorporate this into it. That's what I'm trying to say. So that will be an added value to it. So that's why it's the best way to learn it. So if you are doing that, that's the best to give back to me, being spending so much time to interact with you. No problem, Gagan. Still happy to do it.
We got one more question, ma'am.
Yes, there is one more question. Instead of a small molecule, I wish to perform docking studies with a small peptide. Then how will I get to know the microbial proteins to which the peptide will bind to?
Great. Simran, now you have to tell me what is your small peptide? How many amino acids? Based on that, I'll answer. What is the maximum number of amino acids in the small peptide? I don't know how many of them are there. Let me Google it.
Oracin.
Oh, it's a big protein, right? No. No, this is different. Do you know how many amino acids are there in that peptide? Sorry, I have to go through the papers. Known as personal protein. I can see it's
a protein.
huge. Yeah, it's a protein actually. Oh, okay, yeah. So if it is a large protein, you have to go for protein-protein interaction studies and MD simulations. My old school favorite tool is HEX, HEX. It does protein-protein orientation matching using Fourier transformations. Now you have a lot of online tools like HADDOC and Cluster Pro and all those things. But as I said, old school offline tool is HEX software. But if you're looking at small peptides, 6 to 7 is fair enough. But keep in mind, if it is 6 to 7, your pocket is also big. Unless your pocket is big, you can do blind docking, doesn't make any sense. Unless you know what are the key amino acids you want to restrict based on the peptide docking, what does it make sense? And PPI studies, protein interaction studies, it's also a biased situation. Even crystallographers also complain it, because there you more look at the shape, electrostatics terms of explanation, nothing more than that. So you have to be very careful when you're doing this. So peptide protein, you can do it on CSAR for 6 to 7, but accuracy, it's a question, nothing guaranteed much. So I always recommend protein docking methodology. Which tools are used in molecular docking? Sana, that's what we did today. CSAR, where you can use it for molecular docking for small molecules. And yesterday, Professor Hassan also mentioned about InstaDock or AutoDock Vina. You can use it for molecular docking. What I was telling you about hex, haddock, and clusteral pro was for protein docking. If you're instead of a small molecule or a drug molecule, you are going for another protein, then you have to use another tools which are free. Hex, then clusteral pro and haddock, they can help you with that. Hope I answered, Simran. Thank you. Welcome. I can see many faculties are also here. I'm sure, I hope this was useful to implement it on your research too, for your projects, upcoming projects, so that it can get added value.
Yes, for sure.
And finally, to conclude, if you do not use computational methods in the right manner, in the right place, it is going to fail in your experiments. It is going to increase the cost. It is going to waste your time. If you're doing computational, everything, including the input data, is right, and if you're doing everything properly, every strategy, everything is good, it will be very much helpful to reduce the cost, time, and productivity. So don't think that softwares are going to do wonders. Never it will do until you are expert in it. So science of relevance is very important. Don't think numbers are going to take you to a different level. You must have a good synergetics between the mechanism, the score, energy, interactions, all has to combine together. That makes much more sense. Okay, that's all for now. And I think I'm done.
Okay, so thanks a lot, Dr. Girnath Pillai, and thank you all the participants for being with us in large numbers till the end of the session. All of you will be getting your participation certificates. Only those who attended at least one session on each day will get the participation certificate. And on the behalf of the organizing committee, Department of Microbiology, Ramlal Anand College, I sincerely once again thank Dr. Girnath Pillai for taking valuable time to deliver these two sessions today, which were very, very elaborate, very insightful, and they are going to be very, very helpful to all the researchers, budding scientists for their future work or future research. I am also very thankful to my colleagues who helped me in organizing this particular workshop, 2 days workshop. Dr. Sunila, she worked day and night to make it happen. Dr. Prerna Diwan helped a lot. And we have a team of highly motivated students, which is led by Mr. Anurag Singh from final year of BSc Honors Microbiology, and Pritika Baranwal, Purvi Saini, Rakshita Yadav, who were helping us out throughout these sessions. And even before the sessions were held, they helped us with organizing everything from designing of poster to designing of participation certificates, and for very smooth conduct of both the sessions on both the days. Thank you all.
Thank you so much, madam, for the opportunity. Also to all the organizers. Thank you.
Thank you, Dr. Pillai. Thank you, ma'am.
Okay. So those participants